This is our last panel before lunch, and we're going to focus on the small topic of cancer immunotherapy. And I think we've got a very well-qualified group here to discuss this topic this morning, so I'm really looking forward to the discussion. What we're going to do is I'm going to introduce each of the panelists. They're going to say a little bit about their company or institution and the work that they're doing in the field of cancer immunotherapy. And then we'll follow that with a discussion, and hopefully we'll leave some time at the end for questions from the audience. Before we start, though, I just wanted to observe that one of the things that I think is different about this discussion today, certainly in the context of cancer, and we discussed this during our prep for the panel, is that this is a very different environment. It feels like it. I think many of you heard this in the fireside chats this morning and in some of the presentations that preceded this. Certainly in oncology, I think there's now an appreciation that the immune system and immunotherapy is here to stay. It is something that is, I think, understood to a much greater extent today than certainly it was 20 years ago, and we're now really seeing the evidence that this works, whether it's the checkpoint inhibitors or the cell-based therapies that we're going to be talking about a lot today. And it's really a question of how we build it from here. My view is that this is not going to be a scenario where there's 2 or 3 companies that are going to dominate the field and be the only winners. This is going to be an entire sea change, I think, in the way we view disease and how we treat disease. It's going to involve multiple stakeholders. It's going to involve companies, large and small. It's going to involve medical centers and hospitals and companies like GE Healthcare earlier this morning and others that are going to help with the industrial infrastructure to support the sector. So much of what we're going to be talking about during the panel today is how these pieces fit together. So I'm going to start immediately to my left with Marc Better. Marc is the VP for Product Sciences at Kite, and we're really delighted to have Kite and Marc coming here all the way from California. So,
thank you very much, Morrie. Let me just introduce Kite Pharma to you. For those of you who aren't familiar with our company, we're developing a diverse portfolio of engineered T-cell therapeutic products in the cancer space. We have 2 main platforms that we work on, chimeric antigen receptor engineered T-cells or CAR T-cells, and also T-cells that are modified to include a specific new T-cell receptor, TCR, into their genome to recognize tumor targets. Just by way of introduction, the company was founded in 2009, so we're not exactly a new company, but we really started to gain traction at the end of 2012 when we signed a CRADA with the National Cancer Institute to commercialize the product portfolio that was being developed by Steven Rosenberg at the NCI. That partnership allowed us to start bringing molecules that were already clinically proven through commercial development. Since then, the company has grown quite considerably, and I can talk about that as we go on.
Great. So next up is Elliot Forrester. He's the CEO of Immunocore. Elliot?
Good morning, and thank you very much for the invitation to speak. More appreciated. So Immunocore is, I guess, the immune oncology that's not a cell-based therapy company. What we do is we take the T-cell receptors that Marc has already mentioned, solubilize those, and isolate them for specificity against antigens that are presented only by cancer cells. We then modify those through some protein engineering and deliver them so that the immune system can be redirected to the cancer cells that contain that specific antigen. And we're going to talk about where we are in the clinic and so on and so forth. So the company itself was founded in 1999. We're based in Abingworth, just south of Oxford. We have been working in this space even before people thought it was sensible, indeed when people thought it was madness to be operating in the immune oncology space. And I know the world has changed, and you've alluded to that, Morrie. We raised some money at the beginning of this summer and continue to progress our portfolio on the basis of that.
Wonderful. So now we'll move on to Marc Lowdell. Marc's the Director of Cell and Tissue Therapy at UCL, right up the road, down the road.
And you've got a few other titles that follow that, but is that the main title? Did I get that?
Yeah.
Okay.
So as Morrie said, I'm the Professor of Cell and Tissue Therapy at UCL, and I direct a GMP manufacturing facility. So I came into immunotherapy in 1989 with the first cell therapy delivered in London, and I replaced Malcolm Brenner, who some of you will have heard of, at the Royal Free in 1994. My job is as a translational immunotherapist. I believe that cell therapies and their associates can change clinical practice. And these are still coming out of academia. So I think part of the reason I'm here as an academic is to show you that academia is very busy. UCL as an institution, and just as an example of the UK, not specifically UCL, but UCL is the largest, UCLP is the largest academic health science centre in the UK, and certainly the leader in Europe, if not the world, in cell and regenerative medicine. We've got a budget of £67 million a year of hard-earned cash from research organisations. Just to give you an idea of the diversity, we have our own CAR-T program you're probably familiar with, and a spin-out company called Autolus, developing technology from one of my colleagues, Martin Poulet. And Martin Poulet's technology is behind the Selectis story that you all heard in the press last week. Syncona are very closely involved with us, and we have two new companies, one of which I'm very intimately involved in, will be announced very shortly. My NK cell technology, which I hope to tell you a little bit about later, is already licensed into a US company, and we've gone through phase 1 and phase 2, and we've done the tech transfer. We now have a new company, which I formed 2 weeks ago, which is taking that technology into an injectable. As you know from what you've heard, we have projects across UCL in eye disease, particularly with Pfizer and Robin Ali and Pete Coffey. I work very closely with Vidirigen and the Cell Therapy Catapult, developing translational therapies now in phase 1 and just funded to phase 2 in tissue-engineered regenerative medicine. So things which we would have said 2 or 3, 4 years ago are completely undeliverable. They're just barking mad. We're now delivering them in phase 2 trials across Europe. We did the first ever phase 3 trial of a cell therapy in 2008 with Cellmedica, which is another spin-out. So what perceived by the outside world as too difficult to deliver actually can be delivered. But the important thing from my perspective is that those of us in academia who are developing these have an obligation to make certain that what we deliver in a system that's fit to hand over to these guys to develop commercially. Because me treating 10 patients at a trial at UCL is academic arrogance, if you like, and it's unethical unless we plan to put it in a fit state to go out to these guys to develop clinically. So that, I think, is the sea change that I've seen in the last year. Academics are now believing that we have to do things in a way that is fit for our end-stage delivery and full commercialization of our products.
Great, thanks, Marc. And then last but not least, we have Lothar Germeroth from Juno. He's the Senior Vice President and Managing Director there. Lothar?
Yeah. Well, I'm a new addition to Juno, I have to say. I worked 12 years in smaller biotech.
Right.
So we've been acquired by Juno. Stage Cell Therapeutics was acquired by Juno in May this year. And from there, I take over this position. So Juno at the moment has 10 active programs in 6 different indications, all related to CAR T cells and TCR gene-modified cells, so purely cell-based therapeutics. As others, we started in hematological malignancies. We address non-Hodgkin lymphoma, and the holy grail is whether we can get also efficacy in solid tumors. So that's one thing, and I think the strategy of Juno is really to merge science and production technology. So we have been acquired for production technology. We had a new selection technology, we had new expansion technology, and we automated this entire technology. And therefore, we've been acquired, and that's new. Also with that panel, you see that guys from production like Marc and me are speaking now to investors. That's really a new thing, I think. That was not, I don't remember that before.
So talk to me about that a second. I mean, we're going to cover some of those topics related to manufacturing, but since you mentioned it, and this is an investor audience, or we have investors here today, I mean, so you're being invited into those discussions because they want to understand what the plan is, the manufacturing strategy, especially for autologous therapies, and how this can be done. And so how is it? Are these smaller meetings, or how are you engaging with them? Is it one-on-one discussions?
Well, that's one-on-one discussions. These many of the investors are very well educated at the moment. They knew the, so to say, the drawbacks of these technologies. So cost of goods, supply chain, these are really topics.
Right.
And I hadn't had before, because and the value of those products are really related to those topics. That's clear. So.
You want to think of that more?
No, I'd like to reiterate that. That's definitely my experience as well. So most of the people we speak to are interested primarily in where we are in the clinical strategy, what the results are going to be, the efficacy of the product. But people are sophisticated enough to realize that if you can't manufacture these things consistently, robustly, to meet the market demand and a number of indications, that you really don't have a product. So certainly within our company, we think very seriously about both aspects of development as being synergistic, and we focus quite a lot on, how we're going to commercialize the product. In fact, when I joined the company 3 years ago, that was the main objective of my working there, was to figure out, okay, how are we going to start commercializing these products even before we'd really paved the clinical path. It's matured quite a lot in the last 3 years, and now, I think we're in very good position as a result of that.
Right. So what I think we should do next, because I want to come back to this and pick up on that in a little bit more detail, but I think, again, to set the stage for this discussion, it would be helpful for the audience if each of you could talk a little bit about the clinical outlook, because I think what's going to push this is really the pace at which these products are obviously moving through the clinic, what you expect to see over the next year or two in your lead indications, and then follow-on programs. So, Marc, why don't you start, and then we'll go down the line.
Sure. Just very briefly, our lead indication, our lead product candidate is KT-C19. It's an autologous T-cell therapy against CD19 antigen expressed on both leukemia and lymphoma tumors. This product has completed phase 1 enrollment. In fact, the data from the phase 1 trial will be presented at ASH at the beginning of December, so those of you who are interested can attend and see. We have 4 presentations that will be given at ASH on our program run by Kite, and also our collaborators from the National Cancer Institute. And we recently announced that we've started enrollment in phase 2 pivotal trial, assuming that the data support that, in 2 separate lymphoma indications, and those trials are now currently enrolling. And because of the community interest in supporting these trials, we expect that they'll enroll quickly, and we should have data sometime next year. And we're aiming for filing in at least in the United States towards the end of next year, and be able to launch the product in 2017. Beyond KT-C19, we have other programs in clinical development. Primarily some of the engineered T-cell products, which I'll be happy to discuss in more detail if you're interested. They recognize a number of different types of tumor markers, primarily focused on solid tumor indications. And there are various strategies we're pursuing to expand the portfolio to additional CARs and TCR programs.
Right. So, Elliot, if you could describe your pipeline, and maybe some of the partnerships, too, that are...
Yeah, of course. So the organization spent its time figuring out how we find uniquely cancer-related peptides and then the antigens that are expressed in them that are suitable for soluble TCR technology. So that's, I guess, our engine room, which we've been doing for 15 years or so, is developing that, as well as building these modified soluble TCRs. Our most advanced one of those is therefore against an antigen which is called GP100. It doesn't matter too much other than it's expressed in a range of tumors. We have to date advanced in 2 tumor types, both related to one another. One is metastatic cutaneous melanoma, and we're in phase 2 with that program and recruited significant numbers of patients. And we also are operating in a smaller group of melanoma patients called ocular melanoma. And we announced recently that we now have agreed a pathway to market in Europe using the adaptive pathway, which was mentioned in the previous presentation. And we are also, with the same antigen, pursuing another one or two indications that would be of interest because the antigen is present. In addition to that, we have a number of partnerships. So the organization has partnered with some big pharma groups. So we have 4 specifically in the shape of MedImmune, GSK, Lilly, and Genentech. And those partnerships are bringing molecules through to the market. And should our partners choose to announce that, then we'll announce it as well. If they don't, we won't. And we also have our internal pipeline. Again, we haven't talked publicly about that, but there are molecules in different disease settings that we're pursuing. We only go after solid tumors. We haven't as yet tackled any of the liquid tumors because the technology allows solid tumor approaches. And we've certainly published some data, a limited amount of data so far from the metastatic melanoma of the cutaneous variety, and we're seeing some nice results there. The other part of the thing, I don't know whether we want to tackle partnerships per se in the new world, or should we come back to that? Okay. So one of the other things we've been doing, and we all know this, is that we gain a greater insight into cancer biology pretty much every day that passes. And it's clear that tumors adapt from the first onset of the tumor all the way through, and different tumors, the mets from different tumors do different things, and that's why it's been so difficult to tackle it. And so we've taken an approach, and this is now common across the industry, of setting up partnerships to look at combination therapies. And we announced 2 earlier this year, one with MedImmune and one with Lilly, to look at checkpoint inhibition combination and chemokine inhibition combinations to see if we can enhance the benefits of both the checkpoint inhibitors, because we're driving TILs into the tumor, and the checkpoints ought to work with a greater TIL number environment, and of course to look and see if there are other chemoattractants can work as well. And our belief is that, and this is, we were chatting about this in our preamble, is that the new community will be one of collaborations. And what's very interesting, and there may be some big pharma folks in the room, is that big pharma and biotech are thinking differently. So neither side have given up rights. We're, in some circumstances, simply cost sharing, and other circumstances finding other arrangements in order to advance the understanding of the biology and the therapeutics. And there's a kind of, well, let's figure it out when we get there. It's not quite like that, but let's sort of figure it out when we get there and follow the data. So I'm very optimistic about the opportunity for all of us to be able to bring multiple technologies together in order to bring a positive benefit to the patients, but at the same time allow our businesses to advance independently.
Yeah, I want to come back and talk about that a little bit more later too. But Marc?
So, as the academic up here, I've been involved in cancer immunotherapy since the early 80s, and certainly cancer immunology before that. So, I was asked to give an overview to you all as to what the differences are in terms of what's being presented today, and what other areas you'll see and be familiar with, or become familiar with. And I think that the take-home message, first take-home message is that we've already heard that we have one phase 2 pivotal trial. So completely differently to conventional pharmaceuticals, we're talking about small number trials getting phase 2 pivotal data. And that is unheard of, and it's why there's such an interest in the field. And that's not just in CAR-Ts, it's in other things. So CAR-Ts are the big boys on the block at the moment, but we started back in the 80s trying to activate natural killer cells in patients, and I'll talk about that in a minute, because we knew that natural killer cells, which we all have, 10% of our circulating lymphocytes, if you're old as me, it's probably 15%, are there as an immune surveillance package that deals with every tumor that's arising in us every day of our lives. And there's a very good study from 3,765 normal healthy individuals in a single town in Japan, published in The Lancet in 2000, where they just looked at the function of their NK cells when they're fit and well and followed them for 13 years. And they identified that those who had a higher NK activity actually had a statistically significant lower incidence of any tumor. So we are talking about a fundamental response that's going on in all of us, and what immunotherapy is targeting is trying to find a way of curing those cancers where the immune response has failed to target it in vivo. And the immune response is very pleiotropic. It's evolved over millennia. Okay? NK cells in us contain some of the proteins that are expressed on NK cells in earthworms. Okay? Now, earthworms don't have T cells. T cells only appear in about 0.01% of the animals on this planet. But they're fundamentally important in all of us in terms of fighting infection, and they can be targeted to fight tumor. And so the technologies that you're hearing about are CAR Ts, so taking a T cell and changing its receptor, because a native T cell only sees a tiny peptide of a protein, and that protein has to be presented in a way that is unique to you in the tissue antigens that are specific to you. By putting a chimeric antigen receptor T cell into that, and there are many, many groups, including those obviously in my institution, where you can target a whole protein on the basis of what it looks like. So you use an antibody to target that. And that's a very powerful technology. It's certainly been dramatically successful in blood cancers and lymphomas. There are issues about getting those cells into solid tumours, but with the smart technologies around, I'm sure that's not going to be too much of a problem. And I know Kite, as one example, are working on that, and certainly UCL is working on that. You can change the T cell receptor, so you can use an engineered T cell receptor, so it targets a specific antigen in an autologous fashion, so in the patient. And that's what you've heard about and what you'll hear more about. We've been looking at natural killer cells because they're not antigen specific. They target something that's abnormal about a shed load of tumor antigens. So upregulation of heat shock proteins, which is common in tumors, or expression of MIC-A or MIC-B. And we know then that an NK cell will target multiple tumors. It won't just target a specific target, an engineered target that you've put into it. So we're quite interested in that. The problem with NK technology is that you have to keep the NK cells alive and running in the patient. And we've got some novel technology, which is now finishing phase 2, and the data will be unlocked at the end of December. So I can't share those with you, I'm afraid. So got a number of different technologies out there. Before you even start to look at manipulation of cells which are not traditionally immune effector cells. So I'm part of a purely academic, non-commercial MRC program grant for 5 years with £3 million to take mesenchymal stromal cells, the cells that you've heard about for regenerative medicine so much and that we're using in our regenerative medicine platform, to take those cells and genetically modify them so they become cells that can target cancer. And we're doing that in a solid cancer, in lung cancer. That's because when you inject MSCs into an individual, whether they be autologous or allogeneic, they go to the lung at first instance. So if you have a patient with metastatic lung disease, then these cells will go to the lung, and if they carry with them a protein which is cytotoxic for tumor cells, you will kill the tumor cell. We've cured a lot of mice with these cells, okay? And famously, immunologists are very good at curing mice. We're not so good at curing patients, but we're getting there. So I think that probably sums up is that
That was a nice
reasonable?
overview. That's good. That's very helpful. Lothar,
Yes. Our lead product is JCAR15. It's also a CAR T cell against CD19. We will also start pivotal trials on that, also phase 2. And we aim to have it on the market in 2 to 3 years. So that's the plan here. We have in the lymphoma area, we also have very good data. I mean, you probably not know that Juno was based on the programs of Memorial Sloan Kettering Cancer Center and Fred Hutchinson Cancer Center in Seattle. These both very, very good scientific organizations had, so to say, put the programs in. And the Memorial Sloan Kettering program is the ALL, so it's for acute lymphoblastic leukemia. In the Hodgkin space, we have JCAR17. So we see here also about 60 to 70% complete remissions. Yeah? So you would say healing of the cancer. And that's in the hemato-oncological space, but we put really emphasis now on solid cancers as well. But here it's obviously a very different situation. You have tumor stroma effects. You have all these kind of adverse effects you have in the solid tumor. And here you have to think about new means to overcome that. We have a program for ovarian cancer, for example, where we address MAC16. That's a specific antigen. But there we have a concept of of armored cars. So this cell is not only recognizing with its CAR T cell this antigen, in addition it expresses IL-12. Right. And this will give those cells an advantage in the tumor environment. Mm-hmm. So that is the type of thinking we believe we have, in addition to just have a CAR T cell receptor in the solid tumors. We have to have additional means to get efficacy here.
Right. So I think we'll come back, talk a little bit about the pipeline more in some depth, but I want to follow up on kind of the notion. When we have, with Kite and Juno, the two companies arguably, along with Novartis, leading the space with the CAR technology. So, as you are moving into pivotal trials or in pivotal trials, and because presumably there's going to be intense interest in the results of these trials and demand for these treatments, you're kind of forced to automatically start thinking about some of the other challenges that we were discussing earlier, which is, and we've heard this in other fora with the Novartis folks, with the if you make the assumption that the approval will be coming in the near term, how do you begin ramping up to meet this demand without losing focus of everything else that you're trying to do? I mean, I think ultimately, as Marc and others said earlier, I mean, this is what we're trying to do. We want to get these products to the patients. So, obviously you're maintaining your pipeline, but at the same time you're thinking about the infrastructure you need to support it. So, how are you approaching that now in terms of the ramping up that you're going to see? And then we'll talk about some of the investments that you're making in infrastructure to support that as well.
Sure, I'd be happy to address that. So, you have to go back in history 3 years ago. I think we were a very small company. Right. It was a very small company, if it even was together in 3 years ago. Novartis was just getting into this space. There's very little entrenched infrastructure in cell therapy manufacturing, as most of you in the audience know. So we developed a strategy early on of working with contract manufacturing partners to support our clinical development for programs. And in the United States, where we've been manufacturing for the phase 1 portion of our clinical trials for KTC-19 with PCT in Mountain View, California. But early on, we recognized that we didn't want to go down one pathway of depending solely on contract manufacturers or overbuilding ourselves in clinical and commercial manufacturing. We wanted to have a flexible model where we could take advantage of the shared risks associated with investing in one way or another. So we started down the path with contract vendors, but we also have built our own clinical and commercial manufacturing infrastructure. Near our headquarters in Santa Monica, near Los Angeles, we've developed clinical manufacturing capacity that will support maybe 300 doses a year of products in our pipeline, which should be sufficient to manage the clinical portfolio over the next couple of years. Concurrent with that, we also have been building a commercial manufacturing facility located near the Los Angeles airport, which will give us good transportation logistics to many markets in the United States and elsewhere, presumably. That manufacturing capacity, we didn't want to make the mistake of overbuilding, and yet we needed to be able to support commercial launch. So we've built what I would call mid-sized facility that will be at capacity able to handle 4,000 or 5,000 doses per year. The facility itself is a modular facility. It's very flexible. It has individual suites within it that can all make the same product, or they could make separate products. So as our portfolio develops and more products go through the commercial cycle, we expect we can use this as a launch facility for KTC-19. If that market becomes very, very large, which it could be since the clinical efficacy of these products is hopefully going to be really good, then we could build additional facilities. They're not very complicated facilities to build. They're relatively simple infrastructure, and utilities are relatively modest. It's a shell with clean rooms in it. We'll bring automated equipment in to service the capacity as we need to. So we're balancing our own internal capacity with trying not to overbuild, because the marketplace will be a little bit dynamic as these products get approved. And we're going to continue to use contract vendors just to either augment what we can do internally, or for some individual products, maybe we'll completely outsource those clinical production or even commercial. The one thing about commercial capacity, it's very limited right now. And I think some of the contract vendors are willing to work with us, for example, if we want to build something together, and we'll certainly explore that if it makes sense. But right now, the capacity is relatively limited.
So, Lothar, what's your experience?
Juno has built its own facility as well in Bovell near Seattle, and we also are able to make thousands of doses a year in that manufacturing facility. But with our Celgene deal, I mean, you probably have known that, it's again another demand added to that, and we have access to a German facility as well in Munich, where we are located in Germany. So I guess, and we are currently discussing with Celgene how we kind of address the production issue in Europe and in Asia.
So is it well defined within that agreement as to what role they will take in the manufacturing versus what they will pay for?
It's still under debate, discussion. But obviously, the biotech company, we have designed our programs. We have addressed the processes and defined the processes. So I think we will be in the lead in first place to kind of help our partners here to come to the products.
Marc, I mean, is there anything you can add just from your experience in the UK and what you're seeing?
So I think there is globally a challenge for the right number and the right skill set within contract manufacturers. And I think small biopharma companies, we've invented the concept of a hotel manufacturing facility for 2 UK biopharma companies, where they came in and used space out of hours when we're free. And I think that is a great starting opportunity for small companies. There will be developments, no doubt, in CMOs. There's a CMO coming into the UK shortly. And so that capacity will expand. And I think the Kite criteria of having their own manufacturing facility, but working with contract manufacturers to allow global reach, I think is going to be absolutely essential. But what you said was really very important, and I think that's a take-home message. It's cheap to build these manufacturing plants compared to a conventional biologic manufacturing, where you're talking about multiple 25,000-litre stainless steel tanks and a huge plant, where I've worked, and I know the infrastructure investment in that. We're talking about very small investments. So for companies that are investing at that end, that saving at that end can be put into addressing the supply chain issues, which everybody's seeing as so difficult and so expensive. So you're absolutely right. This is unique in terms of medicine, in that for the first time, the biopharma company, or the pharma company in Novartis, has to have a relationship with a hospital to procure their essential starting material to make one of their products. That's never happened before. Right. And the regulatory environment requires a manufacturer such as me, with my royalty-free manufacturer hat on, to have supply agreements nailed down with all of my critical suppliers. Now, when your critical suppliers are 500 hospitals around the world, that becomes completely impossible to deal with. And that's going to be the challenge going forward. And we did it in a small way with 10 hospitals academically across the UK and Ireland, and we were able to do it in 2008. So it's doable. It just requires the mindset and the skill set to do that. And that's where small companies have the ability to do it. And historically, big pharma have challenges in that. And I think that's where we're going to see investment in the future, not necessarily in large manufacturing plants.
So, Elliot, as you're listening to all of this, from your vantage point, I mean, obviously the types of partnerships you were describing are a little bit different. And just for the sake of discussion, I mean, some of the advantages over we're going to be talking about combinations, and you mentioned them, and there will be combinations with many of the companies that are developing these very same products, whether these are also combination opportunities with the cell-based therapies, that you're working with. I mean, that's something you can address. But I mean, as you're having discussions with investors, and I mean, do you have conversations about where the future of the types of therapies you're developing juxtaposed to the cell-based therapies?
No, sure, all the time. I mean, it is one of the key questions that investors are interested in. I mean, from a manufacturing perspective, our story is not half as interesting as these guys. I mean, it's a bit dull in that we...
Dull isn't necessarily bad.
Well, we just, produce massive... We can produce in something as big as this TV about 24 million doses, and it's done in simple fermentation vessels, and there's massive overcapacity for it and so on. And so for us, what we do is in-house, I mean, just to make the comparison, we tend to keep the really cutting-edge research element of the manufacturing, and then everything else just gets outsourced. We have outsourced strategies to make sure we're covered and not all under one umbrella and so on. But in that sense, that's less of a focus for us than it might be for someone who are producing cells. And of course, the investors are interested in that. I guess the key feature that when we were raising funding earlier this year that kept coming up was the difference between tumor types that one could go after, and because that's where they started, the CAR T technologies have tended to go after liquid tumors, and we've tended to go after solid tumors. That's just where we start now. We all, I think, over the next few years will cross over, and so that's been a discussion. And of course, when you get into that, the key then becomes what are the differentiating factors that will ultimately win out for patients and therefore for the company in the marketplace. And that's something that we've been focusing on, in particular with our antigen discovery platform, to enable us to use the antigens that are presented from inside of the cells to differentiate perhaps from those organizations who have been using cell surface targets in the first instance, though I know all of these organizations are looking at modified TCRs as well. And so they're really the key elements. So the manufacturing was a kind of, that's fine, but really it's about differentiating. And if one projects forwards, I think this is where the interesting stuff really begins to appear, is how do we collectively find the right place to get our therapies to patients. And I think the stratification of patients is really where the challenge is going to be. And I don't know whether we want to even touch on reimbursement, but let's leave you to imagine that as an audience. It's really about how we think about that. And so we actively think about where are areas that we think with our technology, its strengths and its weaknesses, we can differentiate from everything else that's either present, because of course there's a long history of working in cancer with all sorts of different treatment modalities, but also with those things that might come. And that's why, I'm coming full circle, I don't know whether you want me to do it now, that's why combinations become very, very interesting as well, and we can come back to that.
But I think the combination models, and I mean, so those are I mean, there's been a lot of attention on those, and justifiably so, over the last few years, and I think the market appreciates the potential of these combinations, and I think the investors can understand it. It does, as you mentioned, beg the question of whether the reimbursement environment is going to be able to support all of these various combinations, because I think that that's going to be a challenge and something we have to work through. But I mean, just before we leave this topic, I think what's interesting though is that the types of relationships that you have with your pharma partners, and I mean, some of the more traditional and those that involve some of the combinations, again, I mean, these are being worked out, juxtaposed to the relationships that the cell-based therapies have, whether it's with Celgene or Amgen or some of the other pharmas that are coming into the space. They're different because you're working through a whole host of issues, Marc, and I mean you and Lothar at Juno, and you're going to be driving the decisions there in terms of how that relationship is going to support that perhaps is still unknown? And is that, I'm just wondering, how do you respond to those questions when investors pose them to you?
So before, let me address that in
Yeah.
this way. So kind of wrapping up what we were just discussing about manufacturing, I think Marc made an important point about these facilities not being too terribly expensive.
Right.
The other thing about them is they don't take that long to build. So from a strategic standpoint, you can be flexible. Now from a partnering standpoint, in my experience, the large pharma is not that experienced in the cell therapy space. I mean, there are obviously exceptions, for example, Novartis, but most people are not that comfortable with it and don't really understand either logistics or the manufacturing or the type of space that's required to get into the cell therapy space. So that impacts how you address partnering relationships. Just as by way of, are we ready to discuss that now?
Sure.
By way of example, at the beginning of this year, we announced a deal with Amgen where it's a relationship where Amgen had targets, but they had no experience with
Right.
engineered T cells. We had the experience with engineered T cells, so we've gotten into a multi-product, multi-year relationship to develop specific CARs against targets that Amgen had invested quite heavily in through some of their BITE programs and others over the
Right.
years. So that's a way for both companies to test the water, take advantage of each other, and bring a portfolio of products to the market, hopefully quickly, quicker than each company could do on their own. Now, with regard to the combination type things, we're taking the approach where we announced earlier this year a relationship with Bluebird Bio, where we're going to co-develop a engineered T-cell product related to the HPV, human papillomavirus E6, where we'll try and target T-cells against tumor antigens and take advantage of their gene editing capabilities. And again, that's a way for us to work together. It's not exactly a combination product, but we'll put two types of technologies together and try and develop a better engineered T-cell product. And then the third example, we recently announced a relationship with Alpine for their transmembrane immunomodulating proteins or TIPS, where we'll, again, in the construct, the genetic construct in the T cells, we'll put additional proteins in there to help the immune synapse and maybe make the therapeutic effect of the engineered product better. Again, by taking technology and applying it towards engineering better T-cell-based therapeutics. I think there's a lot of other examples that we could think about with new technologies of mixing them together and working on strategic relationships where both sides benefit from the products that might evolve.
I mean, Lothar, your partnership with Celgene is a little bit different than that in terms of the role they're playing, or I mean, could you describe it for us?
Well, I mean, you all know that Celgene is very strong with its products in the hematologic oncology area. So I think the strategic decision of Celgene was to add a totally different way to address these kind of tumors with the cells. So it was more, and they demonstrated that they are innovators in the field. Yeah.
Right.
So, and I guess that was their main reason to step into the cell therapy, because they realized that the data in the field are really strong, and they want to be part of that progress in the field. They are very innovation driven, and I have to say that I really admire Celgene in a way that they have kind of conserved their biotech spirit, because they are not for such a long time a big company. So they grow very, very fast over the last, say, 5 years. So, and that helps now in our interaction, I think.
Right. Before we switch over to, because I want to see if we have questions from the audience too, we talked again in our prep conversation a little bit about the regulatory environment and some of the issues we need to think about. And, are we, again, as these products move through the pipeline, and now we get to the stage of registration and approval, and we have to show things like potency, we have to characterize these products. Can we accurately characterize these products? Is this kind of a looming question for the space? Is this something that's going to be a challenge for everyone as we get there? I'm just curious to hear what you all have to say. I know, Marc, you've got some thoughts on this.
Well, I think we are in a very kind space, actually, from a regulatory perspective, both in the US and in Europe. I've had extremely helpful interactions with regulators, and it has been on the basis of a learning curve. And I think we have reined back a long way from where we were even 3 years ago, with quite unrealistic expectations from a regulatory perspective about how we can define our products. But the note this morning that the process defines the product, we must move away from the process defines the product, or we don't get to scale up.
Right.
And our obligation, and one of my great interests, is reducing cost of goods by closing down and automating procedures, because that's the only way that we can make this effective. And if the leukemia world has taught us nothing, it has taught us that there is no single agent that cures leukemia, and that is also going to be true. It's also true of solid tumours. So combinations are going to be out there, and there's going to be combinations of cell therapy as well. These are not going to be magic bullets. I can tell you that because we've never had it before. Why should we have it now? So then if you're going to start combining these expensive therapies, they must be at a cost that's deliverable. And that's really the challenge. And the regulators recognise that. Potency assays, which you alluded to, are very tricky in this field. And as an immunologist, a reliable potency assay with a CV of less than 5% doesn't exist at the moment. But actually, the potency assay, it can be doubly valuable to us in the marketplace because you're using the patient's own cells to make the product. So that potency assay tells you whether it's going to work. That's completely unusual for any drug. Okay, if I prescribe a drug, or when I prescribe the drug, you don't know whether it's going to work. You know it's potent, but you don't know if it's potent in that patient. But because you're testing the patient's own cells and they are the drug, you're actually that much closer to knowing that it's going to work when you put it in the patient. So you have the ability to say, actually, I can't make this, I'm not going to charge for it. Or I make this and it isn't sufficiently potent, therefore we're not going to sell it. So you can reduce the end cost to the marketplace because, as indeed we've seen with novel marketing of some of the anti-myeloma therapies, patients only, the French government's only paying for successes. Right. Unfortunately, they've got rather more successes than they thought, and they're looking at a financial disaster at the moment. But we might see that going forward as a business model, and I think our potency assays will help us in that a long way.
Anyone else want to comment on that?
I'd be happy to talk about the regulatory interactions, at least from my perspective.
Sure.
So we find, it's kind of, as Amy mentioned this morning, we find a very receptive regulatory environment. I've had quite a lot of interactions with FDA related to our portfolio of products, and they are very interested in learning from our experiences and working collaboratively with us. Of course, they have some guidelines that they're going to enforce, and we can't necessarily expect any dispensation because these are complex drugs. They have to be safe and effective.
Right.
But they are very interactive, and I think we're starting to see that now in Europe as well, that the regulatory agencies are interested in learning about these products because they do have this tremendous potential for benefiting human health, and the agency, that's not lost on the regulatory agencies. And they're very interested in our strategies for things like comparability, working with us on design of appropriate clinical trial strategies that will give us definitive endpoints, demonstrating clear benefit. But it seems like it's a fairly open environment, and what we're trying to do is share as much information as we can generate. I think as a small company, it's difficult sometimes to generate the level of information that we'd like to over the short term, but we're committed to doing it, and I think they recognize that and respond in kind.
Great.
I recognize you want to go to the audience, but so I've been developing drugs and taking them to the market since 1991, which is not as long as everyone here, but it's a little while. And there have been peaks and troughs in interactions with regulators, and I would say the denominator is efficacy. So the wave that came out of the 1980s were efficacious and new. The stuff that in the trough was just slightly differentiated from that efficacious stuff. And we're in a whole new world. As you opened up right now, 70, 80% efficacy rates, it's about efficacy. The FDA, the EMA, all the other regulators want to get drugs that make, therapies that make a difference to patients, And that's what it's about.
Yeah, absolutely. So we've got a few minutes left. I'd like to open it up to the audience. So there's a question back here. waiting for a mic here. That one on?
Can you?
There we go.
Now you can
You're
hear
live.
me.
Thanks very much. Marc alluded to the recent and tantalizing data from Selectis around allogeneic CAR T, and I'd be very keen to hear what the views of the panel are regarding whether this is the next step change in this exciting field.
So would you like me to kick off on that?
Sure, why don't you kick?
So obviously, there's very little I can say about it. I wasn't personally involved. I know the technology behind it. Allogeneic therapies are always terribly interesting to the world because there's this panacea that they're off the shelf. But you still have to have a donor. So it's not entirely, it's not like a conventional pharmaceutical. It has all of the benefits of being more quickly available, but one's not, it comes down to a cost of goods issue, again, as all of these things. If it is truly off the shelf, you've still got to keep it in manufacture. So you've got to manufacture these things and store these for a marketplace that doesn't exist yet. So effectively for your trials, you're making enough because you know that you're going to be able to do them. But you're going to be driven, and I don't know the business plan for Selectis, and I wouldn't pretend to guess it, but I know what the problems that I would face if I were trying to manufacture it in my unit, which would be how many do you manufacture, how many doses do you manufacture, given the cost of manufacturing, how do you show the stability of that product? And we're still talking about a chimeric antigen receptor where we have a very limited number of antigens at the moment that have been, CD19 is the only one that we can definitely target. Whether it will be better than an autologous CD19 is still out. That's got to be proven by the clinical trials. But one great patient is one great patient.
Just, I don't know whether Juno and Kite want to comment on the allo outlook as well.
So I just read the same things probably that you read last week about this remarkable case. And it's very impressive. I think the time will tell whether these things have the clinical benefit compared to autologous therapy. And at the end of the day, while I think the cost of goods is extremely important, and all of us are working very hard to keep that down as low as possible through things like automation, streamlining manufacturing processes, it comes down to efficacy in the clinic and potential side effect profiles of these allogeneic therapies. You know, if the innovators are really successful in eliminating things like the autoimmune response or other bad potential side effect profiles from these kinds of therapies, then they will find a place in the market. For now, I think it's very tantalizing. It needs to be demonstrated efficacious.
So you all are, go ahead.
Just want to make a point. The unique thing about all of these therapies, most of these therapies, is that the dose you give is not the effective dose. Okay, so these cells proliferate in vivo and they stay in patients. We've shown that with our CMV immunotherapy years and years ago, and it's true of CAR T in the patients who've had them now a long time out. Whether that's true of an allogeneic product, leading to long-term engraftment and maintenance of immune response, because these tumours are not cleared in one hit wonder, they do come back. So it may well be that the allo story and the immunogenicity of the allo product, even though it's been reduced considerably by the genetic engineering, may indeed be a long-term problem where the autologous product will engraft and stay in that patient potentially for life. I think we've had 10 years. First HIV CAR T cells now have been shown to be in the periphery 10 years later.
Wow. Lothar, I mean,
I think it's an interesting approach, definitely, but it's one patient, and we have to look. I mean, they use heavily campus that will obviously have other side effects, like be prone to infectious diseases and others. So I guess we have to see more patients to kind of evaluate the side effect profile versus the efficacy.
Mm-hmm. Other questions? We have time for maybe one more. Anyone else? There's one over here.
I think this was alluded to earlier, but I think I'd like to return to it. Is some of the IP issues around who actually owns some of the CAR T therapy still a little bit hazy? And some of the
do we have any lawyers on the panel?
Are the patent lawyers going to be some of the biggest winners out of this technology?
So I told them they wouldn't have to answer any questions on IP before. I mean, I don't know if you want to duck the question completely. I mean, obviously, I mean, is there anything you guys can offer just from a high-level perspective?
I'll tackle it, but I won't go too deep on this, if you'll allow me. So there are patent estates surrounding the particular targets in many cases, whether it's the antibody portion of the CAR or the TCR itself. I think in the United States there's a reasonably clear patent estate around some of the CAR technology. Don't believe there's much protection in Europe around the TCRs. I think there's very specific patent estates, but it's varied, what it comes down to.
Well, I would say the CAR is only one portion of the entire process.
Right.
You should not forget that. Yeah? So the strategy of Juno is obviously to have a solid IP portfolio around the CARs, but in addition, we really look on cell selection, on automation, on cell expansion, and to cover the entire process by having a good portfolio. So that would be mine.
I think that in this space, whether it be CAR T cells or TIL cells or any of the other immunotherapies, or even DC vaccines, which we haven't spoken about today, but
right.
you know, the largest number of patients treated with an immunotherapy so far in the world have been treated with DC vaccines, and we've got big companies now in seminal phase 3 trials, so we shouldn't forget about that. But the thing comes down to the know-how of the manufacturing, and no one will IP that because they have to declare it. Know-how is going to be really, really important in this space, and investors that can spot know-how and the value of the know-how are actually going to be the smart investors in my mind.
Elliot, do you want to talk about your uncontested patent estate?
Thank you.
No issues with the IP for you?
No.
Okay. Good.
Not so far.
Well, I know that ending the discussion with IP before lunch builds up the
Yeah.
appetite. So, but anyway, I think that's it. We've got lunch as next, and I think it's one flight up. So you go out and go up the staircase, and we'll be serving lunch. But I want to thank our panel for a great discussion. Very informative. Thank you.